All right, the last, well, we got two major things here, okay? We've gotten the part where we made ATP and NADPH, okay? We talked about the light-dependent reactions, which happen in the presence of light. Well, now I need to talk about this thing called making carbohydrates, okay? And that's what this is. This is the Calvin cycle, all right? This right here is the Calvin cycle. This is where we utilize now, because we've utilized solar, excuse me, and we've utilized water. That third major thing that we needed for photosynthesis was carbon dioxide. Again, coming up through the stomata, opening on the underside of the leaf. All right. So now we're going to fix that carbon dioxide. We're going to fixate that. We're going to utilize that to make carbohydrates, the Calvin cycle. All right. In much more advanced biology classes, this type of photosynthesis where we are utilizing carbon dioxide under normal atmospheric conditions is referred to as C3 photosynthesis. And it's called C3 photosynthesis because we get a 3-carbon molecule. That's all, okay? But C3 photosynthesis is what we've been describing. Normal photosynthesis. Normal photosynthesis. Okay. Normal. Okay. Those are 2 words there. Normal photosynthesis. All right. Beautiful day. It's nice and sunny outside. It's not too dang hot. I got a little bit of water going on. It's rained relatively recently. Okay. So it's not dry. It's not arid. All right. This is normal Midwest weather. Okay. Normal Midwest winter, let's think of it, or weather, let's think of it that way. Okay. So Calvin cycle is a cyclical set of reactions that involves 3 basic steps. I've got to fixate it, which means I got to attach stuff to carbon dioxide. Then I got to reduce it. Then I regenerate it. And this is cyclical. So your end product is going to be the first step in the reaction. And it constantly just recycles itself over and over and over again. Okay. Okay. So let's look at this thing. There are some shorthand here. These words, okay, ribulose bisphosphate, 3-phosphoglycerate. We'll see glyceraldehyde 3-phosphate here in a second as well. These are all listed on, I believe, the next slide. Okay, so RUBP, and I'll let you use the shorthand if you want to do that for this exam. Okay. Specifically ribulose 1,5 bisphosphate, but who's counting? So that CO2 comes in through the stroma, through the stomata into the stroma. That is where we attach it to ribulose bisphosphate. It's a 5-carbon molecule, so 5 plus 1 is 6. Easy math. You don't need a calculator for that one, I hope. So now I have this 6-carbon molecule. Why do I add 5 to the 1? Because now I have 6, and I can divide that equally in the half into 2 3s. Okay, so the first step is just adding ribulose bisphosphate to carbon dioxide to give me a 6-carbon molecule. Now I can easily divide that thing right down the middle and give me 2 3-carbon molecules, and that's your 3-phosphoglycerate. You can see we have an enzyme that can speed it up. I don't know if I'll ask it, but if I was to ask you to describe the Calvin cycle, if you wanted to maybe add a little extra juice to impress me, maybe I'd look kindly upon that. But I'm not worried about RUBP carboxylase. okay, or Rubisco, right? There are enzymes. We have enzymes for every damn reaction in the body, whether it be us or plants. So it would stand to reason that I would have an enzyme there as well, right? So we have enzymatic reaction that can speed this thing up. But now I've basically fixed carbon dioxide, okay? I've added something to it, okay? And that's gonna allow me to get what I need out of this thing, carbohydrates. Okay? And there's the words. Okay, there's the words. All right, all of this Calvin cycle is happening out in the stroma. Okay, I go from this thing, ribulose bisphosphate plus CO2 to get me a 6-carbon creature, 6-carbon molecule. I immediately cut that thing in half into 2 3-carbon molecules called 3-phosphoglycerate. As soon as I form 3-phosphoglycerate, and this is just showing you the chemical reactions here, and I apologize if that's really bad. It's bad on my screen too, so it's the crappy book's fault, okay? So I apologize. Okay. As soon as we form 3-phosphoglycerate, I break it down, I reduce it to what's called bisphosphoglycerate, BPG. BPG gets immediately reduced into glyceraldehyde 3-phosphate. Why is glyceraldehyde 3-phosphate important? Because it is at this point right here that I can make anything in the world that I want, because I'm the plant. Okay? This glyceraldehyde 3-phosphate, depending upon what the demands are for the cell, for the plant, can make a number of different things. Okay? And I think I have a slide that kind of shows off some of that jazz. All right. To take G3P and manipulate it to form carbohydrates, which give me all my various fancy pants stuff in a plant, I need energy. Where in the hell did that energy come from? From the light-dependent reactions, ATP, NADPH, all right? This is what allows me to make much more bigger molecules. To go from small to something big, I need an input of energy. Where does that energy come from? From the light-dependent reactions in the thylakoid membrane. That's why we talk about it first. So now I've made my carbohydrate. Okay. What does that carbohydrate become? Well, I'll tell you in a second. Okay. But that glyceraldehyde 3-phosphate becomes carbs. Okay. Carbs, carbs, carbs. My carbohydrates. Okay. Ta-da! Calvin cycle has done its duty. Its duty was to make carbohydrates. So now I got to recycle, okay? Because I've taken that, harnessed it, made different carbohydrate structures. Well, I need to regenerate, okay? I need to regenerate that ribulose bisphosphate, okay? So the remnants that remain here are used to regenerate RUBP, ribulose bisphosphate. Okay. I got to regenerate it. If I don't, Calvin cycle can't occur. Okay. So I need this thing. And you can see it takes several turns to regenerate RUBP. Okay. For every 3 turns of the Calvin cycle, that's how many we need to be able to regenerate this thing. So it's not a 1-for-1 ratio. Okay. It's not a 1-to-1 ratio. And again, what's funding this ability to regenerate this RUBP? It's energy, okay? It's going to utilize ATP that we got from the light reactions to help fund our regeneration of RUBP. And that's what you see here, where we fixate it. RUBP is added to carbon dioxide to make 3-phosphoglycerate. 3-carbon creature. We go from the 3-carbon creature, to 3-phosphoglycerate. 3-phosphoglycerate to bisphosphoglycerate. Bisphosphoglycerate, I reduce to glyceraldehyde 3-phosphate. Ta-da! And then I've made whatever glucose structure, whatever carbohydrate structure I need. Okay. Then I have to put in a few more turns here, 2 more, plus this one, 3. And I have to put in a little ATP. Again, I got that from the light-dependent reaction. And I can regenerate ribulose bisphosphate. And the menagerie can continue again. Let's watch a little preview here. Let's watch a little video, see if we can visualize this a little bit easier.
When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Calvin cycle reactions. The Calvin cycle consists of a series of reactions that reduce carbon dioxide to produce the carbohydrate glyceraldehyde 3-phosphate. The cycle consists of 3 steps, the first of which is carbon fixation. In this step, carbon dioxide is attached to ribulose 1,5-bisphosphate, resulting in a 6-carbon molecule that splits into 2 3-carbon molecules. The second step is a sequence of reactions using electrons from NADPH and some of the ATP to reduce carbon dioxide. In the final step, ribulose 1,5-bisphosphate is regenerated. For every 3 turns of the cycle, 5 molecules of glyceraldehyde 3-phosphate are used to reform 3 molecules of ribulose 1,5-bisphosphate. The remaining glyceraldehyde 3-phosphate is then used to make glucose, fatty acids, or glycerol. It takes 2 molecules of glyceraldehyde 3-phosphate to make 1 molecule of glucose phosphate. Thus, the Calvin cycle has to run 6 times to produce 1 molecule of glucose. These molecules can remove their phosphate and add fructose to form sucrose, the molecule plants use to transport carbohydrates throughout their system. Glucose phosphate is also the starting molecule for the synthesis of starch and cellulose.
And again, that's why Calvin cycle is so important, okay? Because now I can make all the stuff that goes into a plant, okay? The stuff that makes up the oils of the plant, the stuff that makes up the simple sugars that the plant's going to utilize to fund itself. Fructose that goes into the fruit that it's going to produce. Starch and cellulose that go into the cell wall. Okay, because when we look at plants, there's actually different types of cell walls. Okay, amino acids, which help to make protein structure, which go into cellular structure, cell wall. Okay, so that G3P, that glyceraldehyde 3-phosphate, goes into a number of different ways. It just depends on what I need as a plant.